And once the gel solution is just thickened, then you put a cover slip on that. And in another slide, you put another slide on as a protective layer or as a cover, and then you put another solution in a petri dish, and that solution just diffuses into the gel, and small crystals are getting developed. And the development of crystal you can see under the microscope. If you have a digital camera facility attached to the microscope, you can record it and you can take photographs. So the method is described over here, and the photographs we have taken, that is on a brushite crystals, and the inhibition we have studied by using a lemon juice or a citric acid or citric acid. So the growth inhibition in vitro is carried out using the citric acid as an inhibiting solution, using a very rapid and simple method, because this is a very rapid method, very simple method. In a gel growth method, it takes almost 15 days crystals to grow. And if you want to study the growth dissolution, again, it will take a few more days. So within 1 month, you can have a complete data that how much efficient the growth dissolution is, growth inhibition or growth dissolution is taking place. But here, the growth of crystal is taking place very rapidly under the microscopic condition, and using the inhibiting solution, whether the inhibition is occurring or not, you can check it very rapidly. Within 2-3 days, you can have a data with you. So this method is very important, and the paper was published in Current Science with Vaidya sir, and it was published in 2007. And the photograph of crystals appeared on the cover page of Current Science. Another is growth inhibition of calcium oxalate monohydrate crystals. And this is done by a very enthusiastic pharmacy faculty member from Pakistan, that is from Karachi. And he has adopted our technique, which I have reported or I have just discussed in the previous slide. He used that particular technique, and he studied growth inhibition in using the glass slides in a gel-based medium. And he used 3 different seeds or pulses, and the name of pulses are given. And these pulses he has used to study the growth inhibition of calcium oxalate crystals. And these pulses are very common. We use in our day-to-day life. So this technique is used by many people. Then how growth inhibition is occurring? In vitro studies, different herbal extracts of medicinal plants are used in a crystal growth experiments. In gel growth, these extracts are added on the top, and the growth rate of the crystals is observed. And aqueous extracts or alcoholic extracts are used. And these theories are discussed by S. Ahmed et al. And in his paper. So this technique is very, very efficient technique. Now I'm coming about the reverse pharmacology. That is the favorite subject of Dr. Ashok Vaidya. So I'm not a competent person to describe or discuss about that. But I will just observe few things about that. If the crystal length decreases, then it is called dissolution, which can be used for the treatment or cure the problem. If the crystal length increase becomes slow, then it is known as inhibition, and that can be used for the treatment to give relief. This thing is very important from a reverse pharmacological point of view, because Sir has always advocated for the reverse pharmacology approach. There are large varieties of herbal extracts available. Their toxicity, their potency, they are well documented. If we use those herbal extracts for pilot study in vitro, that can be very much helpful for paving a path for in vivo studies or animal studies. And we had one project with Vaidya sir and Mengi madam, and we completed nearly 15 years back. And that project we worked in a tandem manner. We studied the in vitro thing using the gel-based model. And then Sir studied in vivo and animal model things. And we came with a very good results in our DBT major research project. The herbal extracts, we used herbal extracts of Tuberous terrestris and Virginia regaluta, and these are used for inhibiting calcium oxalate monohydrate crystals. Again, the photograph is shown about the Tuberous terrestris and Virginia regaluta, and growth inhibition study was carried out by Dr. Vimal Joshi. Right now, he's a principal at Science College in Petlat, Gujarat, and he has grown the calcium oxalate monohydrate crystals in a test tube, and he used these herbal extracts to study the growth inhibition of these crystals. And the paper was published in Journal of Crystal Growth, and that is to be a very important paper. Growth inhibition of Struvex crystals. We used citrus indica lean juice, and significant growth inhibition was observed. And that proves citrus inhibition theory. So citrus inhibition theory, in a simple manner, one can say that if in urine the concentration of citric acid increases, there is a more probability of calcium ion to bind with citric acid, forming a water-soluble calcium citrus salt. But if the amount of oxalic acid is increased in urine, then the probability increases of calcium to bind with oxalic acid or forming a calcium oxalate, which is a water insoluble one. So here the citric inhibition theory advocates that somehow if we increase the citric acid level in urine, there is a less probability of forming a calcium oxalate salt. And this paper was published by us, Dr. C.K. Chauhan, right now working as a faculty in a government science college, Gandhinagar. And we published this paper in 2008 in Urological Research. Then growth inhibition of cholesterol crystals. The cholesterol crystal growth inhibition studies was carried out by using Phagonia critica lin. And this herb is grown widely in Saurashtra University campus. So with help of bioscience department, we identified the plant, and from that plant, we got the extract using the facility available at bioscience department. And using this extract, we got a very encouraging result for inhibition of cholesterol crystals. The growth of cholesterol crystals is occurring in test tube in form of needles, and one can see in the test tube, the needles are grown. These are the cholesterol crystals. So the growth inhibition is occurring using the Phagonia critica lin. And this result was included in the PhD thesis of Dr. Purvesh Vyas. Right now, he's a faculty in Science College in Amreli. The role of tea in urinary stone. So we always prefer tea in morning and afternoon, and it is a common drink in our country. So tea is having a component which is very much helpful in urinary crystal inhibition study. So the effect of minus epicatechin, a component in green tea, is an precipitation and growth of infectious urinary stone amorphous precipitations. So this component, epicatechin, is an antioxidant, and that is responsible for the amorphous urinary stone inhibition. And this study was carried out by a scientist from Poland, Joltana Priver, and she is working on growth inhibition of various crystals. and she is using a software. By using this particular software, she tries to predict that whether this inhibitor is a potent inhibitor or not. And usually for any chemical reaction, the potential energy should decrease. And that is the law of nature. So if two different ions or different active compounds, they form a complex, then if theoretically the potential energy reduces, then it is a potent inhibitor compound. So like that, she calculates the potential energy by using a software or computer program. And she predicts that particular inhibitor is a potent inhibitor. And that forms a good complex. And by that, she has selected several compounds. And in that, she has taken up the component of a T. And that is used for studying the growth inhibition of urinary stone. Some of our research publications, the one publication is took place in Journal of Crystal Growth in 2005. The citation is the highest one, 105, and impact factor is 1.830, and of our group with the Vaidyasar's group. And the next publication is that is in a urological research, and that inhibition and growth of urinary calcium hydrogen phosphate dihydrate using the herbal extracts of Tribulus terrestris and Virginia ligaluta. And the citations are 76, and the impact factor is 1.45, and that was published in 2005. This is the cover page photograph of the crystals which we had grown using the glass slides and cover slips using the normal petri dish. And this is a photograph of a bruchite crystal, and that appeared on the cover page of Current Science on August 19, 2007. Another research paper, Journal of Material Science, Materials in Medicine, and that was published in 2009. And the citations are 85, and impact factor is 4.55. This is the round table discussion with Vaidya sir, another Vaidya sir, Vaidya madam, and myself. So this is a round table discussion. And we had a very hectic discussion on that day in Rajkot. This is a picture of our laboratory, the students working in the lab, and just myself looking into the test tube where the crystals are being grown, and students are discussing with one visitor. Our acknowledgements are, we are highly thankful to Dr. Vaidya sir and Dr. Rama madam. We had a very long collaboration with them, almost 25 years. We published high impact factor journal research papers with a citation over 100. And nearly 5 PhD students completed their doctoral thesis using our collaborative work. And one of my students, Dr. Vimal Joshi, he was the first person to work in this area. And a group of students, Dr. Bharat Parekh, Dr. Chetan Chauhan, Dr. Purvesh Vas, Dr. Kashmira Tank, and Dr. Sonal Vasan. So I'm very much thankful to my students, as well as Vaidya sir and Rama for their help and for their active and they use good for good publications. A concluding remark, crystal growth inhibition study by fruit juices and herbal extracts are very important for preclinical research and important for reverse pharmacology. This is forming important in vitro study It is used by pharmacists for identifying potential herbal extract. This is multidisciplinary study. Thank you very much. Thank you very much. This is the photograph before my retirement in university office. Thank you very much.
Thank you very much, sir, for a very informative talk. It was excellent. It's a new topic that we could hear today, and it was very interesting. Ma'am, we could now take up discussion. So if anyone has any questions, you can please ask, sir.
Meerbai, excellent summary of many years' work.
Oh, thank you very much.
It appears so simple, but it is difficult to collate so many years' work and so many papers with high citations. And I think that we have left it halfway in terms of human usage. And that's why you rightly emphasized in the last slide that reverse pharmacology with these plants, with current demography, other things can be done very well. And that's why you remember we had gone to the next door kidney hospital.
Yes.
I think we had gone there, but it did not work out. But I feel that our youngsters should again, particularly medical youngsters, should particularly explore this further. And I would say that in retirement, you're not retired, but we would rather want to have you as adjunct professor. And that is my request to our research director. And you and Ashwin can guide definitely this more, so it comes to light as a product also. But congratulations for a very good lecture. Wonderful.
Thank you very much. Thank you very much.
Yeah, Shobha, now it's open for discussion. I think
if there are any questions,
I find that still 19 participants. I'm pretty sure if we request everybody to put down their now video, see their faces, it will be a live discussion, I think. Share can be stopped. If the sharing stops, then we request both KHS MRC colleagues as well as colleagues from other collaborative institutes. Can we have, if that goes away, the sharing, then we can have that.
sir, if you could stop the screen share.
I've stopped. I've stopped.
Yeah, okay. Thank you very much.
Hiteshi is there, Nirja is there, Dr. Meera is there, Sharwari is there. Anybody, please, if you have any question. I have some.
I saw Dr. Professor Joseph Birbhai. Is he still there? I would like him to comment at least first. And then any of your students who are now faculty members, if they are there.
I think I have given them the link, and I have put a WhatsApp group message that the talk will be at this particular time. But I think that they may be busy in their duties, particularly today is a working day, and people who are working in colleges, they may have some duties, so they may not have. That is why I suggested Shobha Iyer, madam, to put it in a Google Drive. So whenever they want to watch the lecture, they can watch at their own time.
Yeah, may I invite Dr. Hiteshi Dami. Hiteshi, can you put on your video also, please? How much is Goldstone related to NAFLD? are patients of NAFLD also likely to have more gallstones? Yeah, Hiteshi? I see your name here.
Yes, ma'am.
Yeah. So I would like to ask you a question and then I'll guide it to also the Mihir Bhai. Because as one of the things that we talked about when I talked about 4 mantras from a master, one of the things I said was that what are the things that are left, which Sir has already suggested. One thing is for atheroma, for sure. And that I'm going to discuss later on as Dr. Mihir Bhai becomes active after our invitation that is going to go as an adjunct professor. So that would be good. Now, the question that I'm asking Hiteshi, you, are patients of NAFLD more likely to have gallstones? They do have cirrhosis and the cancer, but also gallstones are more likely with them?
It is prevalent in patients with NFLD. Not all of them, but yes, it is prevalent.
It is prevalent, yeah. Say, Mihir Bhai,
Yeah.
fatty liver is so common these days. Even with a milder BMI high, but the visceral obesity much more, and insulin resistance, they are the ones who have fatty liver. And then the fatty liver, it seems innocuous to many, is not so innocuous. It could be associated with gallstones and later on, of course, with cirrhosis and cancer. So I think that has Biocrystals kurua been thought of for gallstones? Dr. Nutan Nabar is, I thought she was here. Has Biocrystals kurua been thought of for gallstones, particularly for gallstones? Anybody from Ayurvedic is Dr. Ashish Bhatt there? Dr. Nutan was here a little while ago.
Yeah. I'm still here.
Yeah, tell me, has Biocrystals been advocated or being used in Ayurveda for gallstones?
Yes. Kutki is recommended. That is Biocrystals is
Okay. So, Mihir Bhai, as you very rightly
I
ended,
would like to add
yeah,
that Biocrystals kurua is hydrocoleritic. Means whereby it increases the water content of the bile. And so there is something called cholestatic jaundice and cholestasis. And that stasis itself appears earlier in the biliary capillaries. But when larger things happen, they accumulate in the gallbladder, the stones kind of thing. And I feel that one area we had discussed, but we had not taken step was crystals of oxycholesterol, oxidized cholesterol.
Oxidized cholesterol. Okay.
Because
okay.
what damages in atheroma as well as everywhere is essentially oxidized LDL. And that oxidized LDL is the protein which is, it's not only so protein crystallization. We have some colleagues now who are working with us and they are very willing to work on protein crystallization with us. And I feel that we have to get Purvesh's paper out after he recovers from his fracture.
Yes.
But all his thesis is the only not published to my mind. All others have almost published everything.
Published, yes.
Yeah. So what I feel that Biocrystals is a very good example Rama has taken up. And what we need to do is some even in vitro studies about how Biocrystals 1 and 2, because Hiteshi has done her PhD on Biocrystals 2 and Biocrystals on NAFLD, on alcoholic fatty liver disease, in vitro work on liver cells. And but we have not studied bile acids, because I've been telling for a long time that there are bile acids which are derivatives of cholesterol. And so there is a cholic acid, there is a lithocholic acid. Now, lithocholic acid word itself means Litho means stone, as urolithiasis. So the bile acid which leads to stone formation is more called lithocholic acid. And I think some of these things can be done in vitro after a good literature search and what others have done, as well as what is within our capacity to do.
May I request Dr. Shobha Iyer and Dr. Hiteshi, as well as Dr. Nutan,